Cell migration is a fundamental process that underlies various biological phenomena such as tissue development, wound healing, immune response, and cancer metastasis. Understanding the mechanisms by which cells migrate is crucial for deciphering the complexities of these processes and developing appropriate therapeutic interventions. In this script, we will delve into the intricate world of cell migration, exploring various aspects ranging from extracellular matrix remodeling to the biophysical forces that drive cell motility. Join us on this enlightening journey as we uncover the fascinating details of cell migration. The extracellular matrix, ECM, is a complex network of proteins and carbohydrates that provide structural support and guidance for migrating cells. During cell migration, the ECM undergoes extensive remodeling, allowing cells to navigate through tissues and access different compartments. One crucial player in ECM remodeling is matrix metalloproteinases, MMPs, a family of enzymes that degrade ECM components. MMPs are tightly regulated and their dysregulation can have profound implications in diseases such as cancer. Additionally, cell extracellular matrix interactions mediated by integrins play a crucial role in modulating cell migration by transmitting mechanical forces and activating signaling pathways. Cell migration is highly orchestrated during embryonic development and tissue regeneration. It enables the formation of complex structures and the repair of damaged tissues. In development, cells migrate in precise patterns guided by molecular cues and mechanical signals. For example, during gastrulation, cells undergo epithelial to mesenchymal transitions, EMT, and delaminate from the primitive epithelium to migrate to their intended destinations. Similarly, in tissue regeneration, cells need to migrate to the site of injury to facilitate healing. Understanding the mechanisms underlying these processes offers potential avenues for regenerative medicine. Immune cell migration is a critical process in mounting an effective immune response. Immune cells, including leukocytes, must navigate through tissues, find infection sites, and interact with other cell types to eliminate pathogens. Chemotaxis, the directed migration towards chemical gradients, plays a vital role in immune cell recruitment and positioning during inflammation. Various adhesion molecules and chemokine receptors regulate immune cell migration and trafficking, ensuring a coordinated immune response. Cancer cells acquire the ability to migrate and invade surrounding tissues, leading to metastasis, which is responsible for the majority of cancer-related deaths. Cell migration in cancer involves multiple complex molecular pathways, including EMT, degradation of ECM components, and intracellular signaling cascades that promote invasiveness. Understanding the mechanisms driving cancer cell migration is crucial for developing targeted therapies to inhibit metastasis. Cell migration is intricately connected with cell signaling pathways, as various extracellular signals modulate migratory behavior. Signaling molecules such as growth factors, cytokines, and chemokines can activate intracellular signaling cascades that promote cell migration. For example, the PEAK-ACT pathway is involved in regulating actin dynamics and cytoskeletal rearrangements necessary for efficient cell movement. Investigating the crosstalk between cell migration and signaling pathways offers insights into the coordination of cellular processes. Integrins are transmembrane proteins that connect the ECM to the cytoskeleton, transmitting mechanical signals and regulating cell migration. Focal adhesions, which are dynamic structures formed at the interface between cells and the ECM, play a crucial role in regulating cell migration. Various signaling cascades and adapter proteins mediate integrin activation and focal adhesion turnover, allowing cells to dynamically attach and detach from the ECM, facilitating migration. The cytoskeleton, composed of actin filaments, microtubules, and intermediate filaments, provides structural support and drives cell migration. The actin cytoskeleton undergoes coordinated dynamics, including polymerization, bundling, and depolymerization, to generate forces required for cell movement. Regulatory proteins such as Rho-GT posase, actin nucleators, and myosins play crucial roles in orchestrating cytoskeletal dynamics and cell migration. Chemotaxis, the ability of cells to sense and respond to chemical gradients, is crucial for migration in various biological contexts. Cells exhibit remarkable sensitivity in detecting subtle differences in chemoattractant concentrations, allowing them to navigate towards favorable environments. Understanding the complex mechanisms behind gradient sensing, including receptor-ligand interactions, the spatiotemporal regulation of signaling pathways, and cytoskeletal rearrangements, unravels the intricacies of chemotactic cell migration. Cell migration plays a pivotal role in neural development, particularly in the migration of neurons and the guidance of growing axons to their specific targets. Neuroblasts migrate long distances within the developing brain, guided by intricate cellular and molecular cues. Axon guidance is regulated by chemotactic and haptotactic cues, as well as repulsive and attractive molecules present in the surrounding environment. Disruptions in these migratory processes can lead to developmental disorders and neurological deficits. Metastatic cancer cells employ various invasive migration strategies to overcome barriers and colonize distant organs. Collective migration, in which groups of cancer cells migrate collectively, enables their escape from the primary tumor. Additionally, amoeboid and mesenchymal migration modes facilitate cancer cell invasion through the ECM and blood vessels. Elucidating the molecular mechanisms underlying these migratory strategies is essential for devising targeted therapies against metastasis. Epithelial to mesenchymal transition, EMT, is a crucial process involved in embryonic development, tissue remodeling, and cancer progression. EMT allows epithelial cells to acquire mesenchymal characteristics, including increased migratory capacity. During EMT, the cytoskeleton undergoes remodeling, cell-cell junctions are disassembled, and gene expression patterns change, facilitating cell motility. Understanding the molecular events driving EMT provides insights into the connection between cellular plasticity and migration. Cell migration is not solely regulated by biochemical signaling but is also influenced by physical forces and properties. Biophysical aspects, such as substrate stiffness, topography, and mechanical cues, modulate cell migration. For instance, durotaxis describes the tendency of cells to migrate toward stiffer substrates. Migration through confined spaces, such as blood vessels and tissue gaps, requires changes in cell shape and deformability. Investigating the interplay between biophysical cues and cell migration unveils a multidimensional understanding of this complex phenomenon. In summary, cell migration is a vital process with wide-ranging implications in biology. It is involved in development, tissue regeneration, and disease progression. Studying the complex mechanisms behind cell migration provides opportunities for therapeutic interventions and advancements in healthcare.